Our last speaker, our last panelist, is actually one of our newest leaders. And you probably could all agree with me after hearing Dr. Dewhurst and Dr. O'Regan, excellence attracts excellence. And so it is my pleasure to introduce Dr. Ryan Fields, who will be talking about the virtuous cycle of academic medicine. Dr. Fields is currently the Seymour Schwartz Professor and Chair of the Department of Surgery here at the University of Rochester. And he also serves as the Associate Director for Translational Research at our Wilmot Cancer Institute. A surgical oncologist and translational scientist, Dr. Fields specializes in the treatment of solid tumors, including pancreatic, gastric, liver, and soft tissue cancers, as well as melanoma. Dr. Fields began his research training at the University of Michigan and completed his surgical oncology fellowship at Memorial Sloan Kettering Cancer Center. He continues to be, and we hope he finishes his career here. Are you with me? Okay. He continues to be recognized for his leadership in cancer care, research, and mentorship.
Thank you. I realize I'm standing before all of you in your lunch, so I will give you guys some food for thought for temporarily. That's my only dad joke, I promise. It's really, really easy to come in as a new chair and give this talk because I get to come in here and talk about all the great things that I had nothing to do with, but I can take all the credit for within this incredible department, and then tell you about all the new ideas that I have that if they fail, you won't know about today. It'll take years. So this is just an absolute pleasure. So I titled this The Virtuous Cycle of Academic Medicine. I think we rearranged the order a little bit because I hope to take a lot of the themes that Steve and Ruth talked about and sort of bring them into how I look at running a modern department within an academic institution. So I didn't come up with this. This is something, though, that I subscribe to quite heavily. So this cycle, you can really start anywhere, but if we sort of start up here and you develop a center like Rochester is of clinical excellence, and we bring in clinical trials on the research side, like Ruth was talking about. We draw in patients and we build programs. And on the clinical side, if you look at this, the clinical side is really the only thing where we make a profit, where we have a margin. And if we have no margin, we have no mission, because it's that margin that funds the research and the education and what we can do in the community. So the clinical engine is what drives all of this, and it brings in the research innovation. And when we develop a trial or a new program, a new research initiatives, that's what draws the talent. And as Steve said, our mission part of the research mission is to drive and develop talent. So this is what brings students and postdocs and residents from around the world that want to train here. And part of our job, I would say, if we're doing it correctly, is to serve the community in which we reside. And that's the our local area, our region. It's our job to come up with research questions and ideas that are a direct result of challenges or health challenges within our community. And then it just it keeps spinning, and the cadence of this spin and the size of that spin just depends on how much we invest in each area. And I think we can look at this and the successes that we achieve in that cycle within the idea of these concentric circles. So what I would say is, if we have an investigator that starts with a new project, something on the back of a napkin that they start studying in the lab, and they can translate that out into collaborative work where it benefits not just what they're studying, but potentially other cancer types or other disease states, meaning you make a fundamental discovery somewhere and you extend it into other realms. And then can we take that and translate that into a therapy that benefits our community and ultimately the world? And if you look at things that have been done in Rochester, like the HPV vaccine or the biopsychosocial model, those are examples of things that started at one point and now benefit a very wide circle. And I think that's the definition of getting ever better and why we're all here today. I came into a department, and this was the prospectus that they gave me that lists the mission of our department. And I think this rings true to what we're talking about today. We focus on patients. We provide extraordinary care. In my department of surgery, we create new knowledge. We develop future surgeons and leaders across a wide variety of expertise. And what I would say the most beautiful thing of coming into this job is learning and getting to meet some of the people, not only in my department, but around this entire health system where we can collaborate and learn from each other. If we're talking specifically about research and how I look at research within the context of our department, this is sort of a different version of a slide that Ruth had put up with how we sort of look at translational research. So traditionally we would start here and you would say at the bench, we bring a therapy to the bedside. We had great examples of that in the first talk with Dr. Weimer, where we're talking about something that was fundamentally discovered in a lab to directly benefit patients and was turned into a therapeutic. What surgeons are really good at, and Mike Stoner, my chief of vascular surgery, is in the audience and can attest to, surgeons are really good at identifying problems and bottlenecks in the operating room and in our clinic. So we can take the challenge that we see at the bedside and then work with others to develop solutions to that in the lab. And we're at a place here with, for example, a world-class biomedical engineering department, optical imaging group that spurned from Kodak and all the great things that are done here. And those are some of the biggest ways that we can identify bottlenecks in the operating room. We can come up with new devices and design new devices and test new devices or new ways to look at things in the operating room. And so I think this is a great way to look at both bench to bedside and bedside to bench. And I'll give you some examples of that as we go through. So I'm going to transition and give you some examples of work that's being done or has been done in our department that I'm really proud of. I could go on for hours on this. I solicited examples from our entire department and chose a few to highlight the spectrum of research being done right now. So I'll start with one that really gets into the sense of both collaboration and sort of the bench to bedside model. And this is a new grant that was awarded to two of our faculty, Scott Gerber and Darren Carpizzo, looking at immunotherapy in pancreas cancer. Scott Gerber, shown on the screen left up here, is a PhD researcher, a world-class immunology expert that's paired with Dr. Carpizzo, a surgeon scientist. You can see an MD PhD who treats pancreas cancer. And they've come together and partnered to look at a new combination therapy in pancreas cancer that's a direct bridge from work done in their laboratory and is now a clinical trial. And what they're doing is they're combining radiation, a drug that's an immune stimulant, and then an immune therapy to target pancreas cancer. And so they really have to overcome two problems. Pancreas cancer is a really hard tumor to penetrate. It's hard to get a therapy into it. So they've developed this vehicle that's kind of like a smart bomb and gets the therapy directly to the cancer. And they also have to overcome the fact that pancreas cancer is also a very immune suppressive microenvironment. The immune system doesn't really work well in a pancreas cancer, so you have to overcome that. And so they've developed a clinical trial where they take patients who are going to go to surgery for pancreas cancer, and before their surgery, they get a biopsy. They undergo this novel treatment, then they do surgery and a biopsy. And the important thing is it puts patients on the therapeutic. And I'll get back to this a little bit later, but they get a sample of the tissue both before and then the tumor removed afterwards. And they can study and compare how this treatment worked and maybe why it didn't work perfectly to be able to tweak it. And this is one of the things that I would say as surgeons, we have a unique ability to foster that type of research because we're in the operating room with patients, and patients are so universally generous to be able to study their tissues in real time like this in a safe and effective way to advance science. That's one example that I started with because it really is kind of that bench to bedside approach. The other thing that I would tell you is we're talking about something like pancreas cancer. We put patients through very big operations when they are being treated for things like pancreas cancer. And cancer, for the most part, and overwhelmingly, is a disease of patients as we get older. And so we have to ask ourselves, if we're doing treatment for pancreas cancer that involves surgery, we want to make sure we can do it safe and effectively, not only so the patient obviously survives and can do well, but also so they can recover in a cadence that's quick enough and they can get on to their further therapy, whether it's chemotherapy or immune therapy or radiation. And if we look at surgery as a whole, more than a third of all surgery is done in US hospitals in people over 65, and that's just increasing exponentially. And older patients have unique challenges, unique physiology that we have to address to be able to do this. And so Gabby Poles, who's one of the faculty in colorectal surgery, is a leading authority on this. And what she's developing are standards so that through the American College of Surgeons and other groups, we have a direct program to look at safe and effective surgery in older adults. She's been awarded several grants in this space. And what we call this through our American College of Surgeons, our governing body, is a geriatric surgery program verification. And what this creates is clinical excellence. If we look at that cycle of virtuous cycle of academic medicine, this is creating a world-class surgical program, a clinical program that will draw in patients. And then we can further this science because this isn't an endpoint, meaning we haven't figured out how to do this perfectly, but now we're able to study that, say, come up with a new intervention that we think is going to more effectively get patients through their surgeries. There's several verification processes to do this that are really important, much like the cancer center designation, to really put us on the map in this space, and Dr. Poles is leading this initiative. Let's sort of shift gears and look at something related to screening. So, we talk a lot about therapies, but in most disease types, including cancer, and what I'll talk about here, vascular surgery, if we can catch something before it happens, if we can do screening, it's a much more cost-effective therapy, and it's a much better experience, of course, for a patient if we can prevent something. Karina Newhall is one of our vascular surgeons, and she's doing some really innovative work where she's looking at the blood vessels in the neck, the carotid arteries, to try to understand which patients who have a high-risk plaque are actually going to progress on to stroke, because number one, it has to do with how we monitor these patients, but also when we intervene. So as most people know, plaque in the neck arteries, just like plaque in the coronary arteries, can lead to vascular problems, in this case, strokes. And so people, when they have a plaque, they get ultrasounds all the time. Many people may have had them, and it's not that the ultrasound's a big problem, but there's a cost to coming in routinely, the cost to society coming in. And most of these scans, as Dr. Newhall has shown, are pretty stable over time, and the risk stays low, but we're not quite sure how and why and when we should stop screening. Dr. Newhall, because of the leadership of Dr. Stoner and our vascular surgery group here, and the clinical enterprise that they've built, have access to over 30,000 scans and 13,000 patients. So a huge data set to be able to ask and answer questions. And in this case, who actually needs to continue indefinite screening? And so she's looked at blood flow through the carotid artery, not just the plaque, as a metric for this. And what she's shown is that if you have stable blood flow over time, you have a very low chance of stroke or surgery. So now you can use things like machine learning and artificial intelligence to do this work on these large number of scans in order to spot things early and then let us know, okay, who needs more testing, who can safely stop. And the reason that's important, again, is we can tell patients, you don't need to do this anymore. Enjoy your life and stop coming in for ultrasounds. So that's an example, I think, of something where we take a large clinical enterprise and are able to ask and answer important questions in that space. We also have something very unique here in my department of surgery, and that's another division of cancer supportive care. I wouldn't say it's an anomaly, but it's unique. Most departments of surgery don't have a group of PhD epidemiologists and experts in intervention and implementation science within a department of surgery, but it's a huge benefit, and it's a group that does incredible things. And the starting point for a division like this is that we have over 18 million cancer survivors, and that number keeps going up, and that's obviously a great thing. But cancer survivors have, as they live longer, have very, very unique changes to their health. Karen Mustin, who's one of the leaders in this space, just published an article this week showing that patients with cancer have very unique and higher rates of insomnia after their treatments. And we don't really understand why, but there's specific interventions being looked at. And you could sort of, across the spectrum, there may be some cancer survivors in the room. It may be something related to diet, to physical activity. But being able to intervene and support patients after a diagnosis of cancer is the mission of this group. They design studies, and I'll talk about their leadership across the country and some of these study trials they do that are longitudinal over time. They look at very specific cancer-related outcomes like nausea and vomiting and cognitive things. Neuropathies is one of the key areas, neuropathy, especially as they relate it to certain chemotherapy drugs, and then all the other problems listed here, including the sleep problems that I mentioned. They don't just look at sort of big picture physiology. They also dive into the biology of cancer with respect to inflammation and metabolism. And then they intervene with things like exercise and nutrition, also drugs, all sorts of things. It would take me, we could have a whole afternoon seminar to hear about what they do. The group is led by Michelle Janelsons, who's the division chief, and that's a picture of their group here. And one of their signature accomplishments is their leadership in something called the NCORP, which is a national network of community partners. So these are cancer centers out in the community, not necessarily at the university setting. And you can see the dots representing all of them, and they lead this initiative. So it's not that all the work is done here, although a lot of it is, meaning we accrue patients to these studies here. They organize and coordinate in the data center and basically the engine that drives this. And they've done some fundamental work in cancer supportive care under Michelle's leadership. What that creates for me as a department chair is, with their expertise in, say, a dietary intervention, is then, well, maybe we can come up with a dietary intervention to affect a different surgical disease, whether it's cancer or not cancer. So their expertise is a huge benefit and creates incredible collaboration within our department. The last thing I'm going to mention kind of gets at that part on the cycle that looks like community outreach and engagement. And this is another example from our vascular surgery group, and this is reducing vascular disease morbidity via community outreach. The principle here is, as most people realize, peripheral vascular disease, basically looking at blood flow to the extremities. When it becomes an emergency, that's usually an amputation, meaning the blood flow clots off immediately and you have a gangrenous foot or a toe. And that's not only a huge burden to the healthcare, but it's and oftentimes a preventable situation or something we can get at earlier. So what Alaska Pendleton, who's one of our researchers with a master's in public health and expertise in this space, has done is she's worked with URMC as well as 2 community organizations to increase our ability to screen for peripheral arterial disease, especially in high-risk populations. So rather than have someone come in with an acute problem to the emergency room that results in an amputation, what she's working to do is get out into the community with these health ambassadors and into various community organizations, whether it's places of worship, barbershops, all the places where people are, to develop partnerships and trust within our medical community and our medical center so that screening could be done and we can know who's high risk and could be intervened on in a more controlled setting rather than result in limb loss. There's a paradigm for this in the cancer space, the idea of these mobile mammography units where we bring mammography to the community. This would be the same concept that Dr. Pendleton's working on to bring vascular screening to these at-risk communities. Again, it creates academic opportunities to study this. It creates ability for our trainees to be exposed to the community and learn how to do this type of very impactful research. So I'm going to shift gears for the last couple minutes and just talk about why I think as surgeons we have a unique ability and unique responsibility to contribute to research. And that's because we have, as I alluded to, this unique access to tissue. Some people would call it biobanking. There's other ways to sort of look at it. And it gets at another version of a slide that Dr. O'Regan showed earlier related to personalized medicine. And that's that what we don't want to do is take everybody with a particular disease state, let's again say breast cancer, and put them all on the same pill and realize that, sure, there's going to be a population where it works, the light blue. There's going to be people where it doesn't work very well. But the worst is we're going to take patients where it doesn't work well, but we're also going to hurt them with the side effects. This is more the paradigm we're going after, these personalized treatments where we use genetic testing or a blood test or whatever it may be, a biomarker, to then decide who's going to go on to what treatment. That's what we want to do. That's what we're moving towards. And so how do we do that? Well, I'm going to show you a slide from some of my work. If this makes you dizzy, it's meant to. It makes me dizzy as well. But this is a diagram, and we'll just focus on one spot. We'll focus on the upper left, where what we did is we took multiple patients with colon cancer. And because we were surgeons and, again, had access to this tissue and could ask the question, what we wanted to know is how is the primary tumor that started in the colon, and that's shown here in the red, different from the metastatic disease when the tumor had spread into the liver? What changed? What led it to spread? So each of these dots in the liver from this one patient are 3 tumors that we collected at the time of surgery, along with their main tumor shown in red. And the 3 little dots there mean that we actually sampled that tumor, that colon tumor, in 3 different spots for this patient. In some of the patients, like you can see this number 10 here, we were able to sample it 7 times. And then the green is interesting, where we took a piece of the tumor and we grew it in a special mouse, a so-called xenograft, where we can then study the tumor in the lab, and we're able to do this in several of the patients. And what we did is we subjected all of these tumors in a very large study, hundreds of samples, to some deep genetic testing. And this is kind of, again, the dizzying results that we found. So the reason I'm going to walk you through it is it kind of gets back to what Dr. O'Regan was mentioning about this sort of idea of treatment resistance and how we develop new therapies. So under the red here are the 14 regions that we studied of the main colon tumor in these patients. And all you have to sort of realize is these different colors and the width and the length of these mean that each of those spots, each of those 14 spots look different under the microscope. It was if they had 14 different cancers growing within one tumor, this idea of tumor heterogeneity. And the reason that that was important was because if we look at a particular mutation called KRAS, which is kind of like HER2, it can dictate therapies in colon cancer, much like HER2 does in breast cancer. You had to look really, really hard, really, really deep beyond what you would normally look like in a patient to find a couple of these spots that actually had KRAS mutations. But according to the clinical test that the doctor ordered on this patient, they did not have a KRAS mutation. So the patient was treated as if they didn't have that, but then over time, treatment resistance emerged. And it was like the one or two little bacteria that had a resistance to the antibiotic sort of started to grow while the others die is what happened in this patient. So only through these types of very deep genetic testing and some of the advances we've made in genetic testing will you find things like that out. And the only way you find that out is by having surgeons and scientists that work together and get this tissue in a way that we can study it, work with patients, as Steve said, communicate to patients the importance of contributing this, because this is what leads to advances in the field. It doesn't have to just be cancer too, and I'll sort of explain why I think Rochester is such a unique place to do this type of work. I don't know how many people read this book. This was a very popular book several years ago called Lifespan that was really talking about aging. And as Dr. Dewhurst alluded to, just the concept of aging, both at the organism level, the mole rat or the human being, but also at the cellular level. And one concept within aging is this idea of senescence. And at a cellular level, it's really the loss of a cell's power to divide or grow. We have cells in our body that stop growing. Like people have called them zombie cells because they just sort of sit there. But now when we've started to study them, they sit there and they release a lot of chemicals, a lot of proteins that are very inflammatory. So they're not doing what they should, but they're not dying. They're not turning into cancer per se, but they shouldn't be there. And these senescent cells are a very popular area of study right now. And what the NIH realized was that we didn't really understand these very well, these senescent cells. Where did they exist in the body? How did it change over time? How is it different between males and females or diet? All these sorts of questions. So they launched an initiative to try to really create an atlas or map these out. So we sat, when I was at WashU, we sat there. Now we're sitting here at Rochester saying, well, as surgeons, we collect all these tissues from the operating room for cancer. Why couldn't we do this for non-cancerous tissues to study senescence? And that's what we started to do at WashU with two collaborators on the scientific side. So again, showing these clinical and research collaborations. And we're able to take some of these real powerful analytics, what we call high resolution cellular imaging, where we can really look and combine the genetics at the cellular level to try to map this out and understand it. And so as an example of sort of what we find when we do this type of work, one of the tissues we were looking at was liver, because we operate in the abdomen a lot, and the liver's just sitting there looking at us, so we can look back at it, take a little piece and study it and look at senescent cells in normal people. So when we did that, we found, again, with these very high resolution cellular imaging, that we find 4 unique senescent populations, 4 different cell populations in the liver, and that they change over time. And one of the most fascinating findings was that it changes with age, but we had a unique group of patients that we studied that were cancer-free, but at one point in their lives before they were undergoing surgery for whatever it was, had been treated with traditional chemotherapy. And when we looked at these large number of patients, we find that chemotherapy, although it of course works on cancer, it causes the liver to age. So if you've been on chemotherapy and you're doing fine 5 years later, 10 years later, your liver just looks like it's about 5 times older than it should be based on your chronologic age. And it has something to do with the way these senescent cells are affected by chemotherapy. We're just sort of at the tip of the iceberg and even understanding this because we've just started to describe it. But this, again, is what creates the raw materials where we can then and investigate this and determine what implications it might have on human health and disease in general. Senescent cells are all over the body. They affect cardiovascular disease, Alzheimer's disease, my disease affliction of male pattern baldness. There's lots of ways that you can sort of get it where this is going to advance human health and disease. So I'll come back to this because I think, again, what I'm trying to show you are examples of how this engine can continue to turn where with an investment at any cycle. I didn't specifically mention the educational prominence, but as Steve alluded to, we put money and we need to put resources into some of the training grants that we look at to attract and train the best and the brightest. One of the greatest things about here are having people like Dr. Mayadas, our new chair of immunology and director of the Immunology Institute. We have the Aging Institute that was mentioned, environmental studies to look at things of environmental exposure and cancer and disease risk, Center for RNA Biology, all these collaborative institutes that work together with the clinicians and the clinical researchers to drive innovation. So I'm going to stop there. This is a picture from a waterfall in Iceland from a trip last summer that is one of my favorites. So thank you all for your attention.